everyone, how are you? I hope you're doing well. Welcome back to our channel. We're still continuing with our series of cell communication and cell division. So we concluded the cell communication series. We discussed the various things that were there. If you've not yet watched them, please do so. Today we are beginning a new topic, and that is the cell cycle, whereby we shall look at the cell cycle itself, the cell cycle. We shall look at its regulation. Then we shall look at cell division. That is mitosis and meiosis, and their overall significance. That is what we are going to be seeing. Let's begin with the cell cycle. The cell cycle, as you hear, a cycle, something is continuous. The cell cycle is a series of events. It's a series of events from one cell division to another, leading to the duplication of a cell into two daughter cells. So what we are going to discuss are the activities in between one cell division to another. The cell does various things in order to prepare for that, and that is what we are going to discuss today. So the cell cycle is made up of two parts. Number one, it is made up of what we call interphase. Then it is made up of what we call M phase. And the M phase is what we call the mitotic phase. So various scientists carried out various experiments, and they discovered that the cell that just doesn't divide. There are various stops and breaks, and those stops and breaks are in interphase, were given the name as gaps. And there are two gaps in interphase, that is the G1, or what they call the gap 1, and G2, what they call gap 2. And in between these two, we have what they call the S phase. And we are going to see what basically happens in that. Then in the mitotic phase, we have 5 stages. Number 1, we have prophase. Number 2, we have metaphase. Number 3, we have anaphase. Number 4, we have telophase. What we usually summarize as PMAT. Repeat, the cell cycle is made up of two major parts, interphase and the mitotic phase. In interphase, you have the gap 1, the S phase, and the G2 phase, what you call the gap 2 phase. Then within the mitotic phase, you have prophase, metaphase, anaphase, and telophase. Let us begin by looking at the interphase. I said the cell cycle begins with a G1 phase. And what happens in the G1 phase? Number 1, our cell is getting ready to divide, and the daughter cell must have the exact contents as the parents. So the cell begins by synthesizing new organelles. The cell begins by making new organelles. For example, if the mother cell had 2 mitochondria, the cell shall manufacture more 2 mitochondria to form 4 mitochondria. If it had 3 chloroplasts, it shall manufacture another 3 chloroplasts to get 6 chloroplasts, such that the daughter cell has the exact number of organelles like the mother cell. Then we shall have synthesis. We said it began with our G1. We shall have synthesis of proteins and enzymes. Proteins and enzymes. Proteins and enzymes. These ones are the ones involved within DNA replication. Things like DNA polymerases, things like pre-replication complexes, and others. Then if the cell has any damaged thymidine dimers, they shall be repaired. So we shall have repairing of thymidine dimers. Repairing of thymidine dimers. So this is what the cell usually does as it is preparing to undergo division. We said we shall have formation of new organelles from the pre-existing one. In fact, when you read some texts, they call it replication of organelles. We shall have synthesis of various proteins and enzymes that will play a very big role in DNA replication. Things like DNA polymerases, things like pre-replication complexes. We shall later see the roles of all these under our molecular biology series. Then we shall have repairing of thymidine dimers, and we shall see later what role do they play. Then the G1 phase. The G1 phase is the longest phase. It can take up to 80 hours. Up to 80 hours, and as within 8 hours the cell is doing all this. Then from here, our cell shall then proceed to what we call the AC phase. The AC phase. And as I introduced, the AC phase is what we call the synthesis phase. The synthesis phase. The synthesis phase. Within the synthesis phase, all these, especially synthesis of proteins and enzymes, was aimed at preparation for the synthesis phase. So within the synthesis phase, we shall have replication of our DNA. Replication of DNA. Replication of DNA. You basically know that our DNA is found within chromosomes, and within eukaryotes we have 46 chromosomes. So during the synthesis phase, these 46 chromosomes shall be multiplied by 2 and get 92 chromosomes. 92 chromosomes. Such that even after, at the end of the M phase, the mother cell will remain with its 46 chromosomes, and the daughter shall also take the 46 chromosomes. Basically, that is what happens during the synthesis phase. Then from there, our cell shall proceed to the G2 phase. The G2 phase is also known as the gap phase. The S phase usually takes about 60 hours. 60 hours. Then from here, our cell shall produce to the G2 phase or the gap 2 phase. So after having made new organelles, having synthesized various proteins that were played a very big role within the replication of DNA, then repairing the thymidine dimers, then we shall have our DNA replicated or photocopied. Then the cell shall now have to increase. We shall have increased cell growth. Increased cell growth. Increased cell growth necessarily means increase within the levels, increase within the cytoplasm volume. Within the cytoplasm volume. The volume of the cytoplasm shall increase greatly. I repeat, within the G2 phase, we shall have increased cell growth. The organelles manufactured here shall grow to attain a specified size. Then the cytoplasm also shall increase, such that even after the division within two daughter cells, both the mother and the daughter contain the same volume of the cytoplasm. And I repeat, the gap 1, the S phase, and the G2 phase are all part of the interphase. All part of interphase. Then from here, our cell shall proceed to the mitotic phase. The mitotic phase. Our cell shall produce to the M phase. And we say the M phase, say the M phase begins with prophase. Then from prophase, the cell shall proceed to metaphase. Then from metaphase, the cell shall proceed to anaphase. Then from anaphase, the cell shall proceed to telophase over telophase. Then the M phase shall conclude with a certain process called cytokinesis. So let's begin with prophase. Prophase. So, as our cell is proceeding to prophase, it would look like this. This is our cell. This is the cytoplasm. Then this is our nuclear envelope. This is our nucleolus. Then we have thread-like structures within the nucleus. Thread-like structures which are called chromatins. So I repeat, this is our nuclear membrane. This is our nucleolus. These are our chromatins. Chromatins. By the way, let's first talk about chromatins. A chromatin comprises of two major things. Number one, it will comprise of DNA, which DNA definitely we know is coiled in thread-like structures called chromosomes. Then it shall also contain what we call histone proteins. The histone proteins. So the histone proteins coat the DNA that is enclosed within the chromosomes. So our cell will look like this. It will be containing, as we said, 92 chromatins. So now, when the cell goes into prophase, certain proteins are manufactured within the cells that are called cyclins. Certain proteins that are called cyclins, and other proteins are called cyclin-dependent kinases. So these two are phosphorylate. They phosphorylate the nuclear envelope. They phosphorylate the nuclear envelope proteins. The nuclear envelope proteins include things like lamins. It's in the nuclear envelope is made up of certain proteins like the H3A proteins. So all these are phosphorylated. Phosphorylated means that there is addition of phosphates. And when those phosphates are added, they are then degraded. The phosphates degrade them. That means the nuclear envelope will disappear. That is the end product. The nuclear membrane disappears. Then number two, I talked of the nucleus. The nucleus also dissolves. Then number three, here we had our chromatins. The chromatins condense now. They condense to form chromosomes. The chromatins condense to form chromosomes. Why this is important? Our DNA can't be divided equally when it is still in this form. That is why it has to be condensed to form chromosomes. So what we have here is the ultrastructure of a chromosome. This is the magnified structure or the zoomed structure of a chromosome. The chromosome is made up of what you call the centromere. Then a chromosome is still made up of two sister chromatids. Then a chromosome is still made up of a special type of protein which you call the kinetocoa. The kinetocoa. Then the ends of a chromosome, this end and this end, are what you call the telomeres. The telomeres. I repeat, in between the chromosome, we have what we call a centromere. Then at the end of the chromosome, we have the telomeres. So still under prophase, say this is our cell. Now the nuclear membrane has disappeared. Our chromatins have condensed to form chromosomes. The nuclear membrane has disappeared. Then we shall have formation of microtubules. The microtubules, these are microtubules. Remember, as we discussed in our cytoskeleton, under the organelles that make up the cell, we saw the microtubules. The microtubules come from a structure called the centriole. These are our microtubules. And there are two types of microtubules in this case. We have what they call the polar microtubules, and we have what they call the astro microtubules. The polar microtubules and the astro microtubules. Still under prophase, we said the nuclear membrane disappears or disintegrates. The chromatins condense to form chromosomes. Then they shall also have centrioles moving to opposite ends of the cell with formation of the microtubules, or what some texts call spindle fibers. So that is it for prophase. After having looked at prophase, the cell enters the next step, that is metaphase. So meta means in between. So assuming this is my cell. The number one step, the centrioles arrive at opposite ends of the cell. They arrive at opposite ends of the cell. They arrive at opposite ends of the cell. That is number one. Then, so these are my centrioles. Then microtubule development is complete. Microtubule development is complete. We say they are what you call the polar microtubules. These are my polar microtubules. These are what you call the astro microtubules. These ones that attach the centrioles to the cell membrane. Then we say meta means in between. So our chromosomes shall attach themselves at what we call the metaphase plate. They align themselves at what we call the metaphase plate. So the major important process here is the chromosome alignment. At the metaphase plate. The chromosomes align themselves at the metaphase plate. I repeat, the centrioles are at opposite ends of the cell. Microtubule development or spindle fiber formation is complete. Then the chromosomes align themselves at the metaphase plate. The metaphase plate is something imaginary, doesn't really exist, but they align themselves there, and we are going to see the significance of that. So from metaphase, our cell shall then proceed to anaphase. Just remember, meta means in between, ana means part, apart. So after micromers, then on top of that, if I forget, the kinetocoa. the kinetochore. The kinetochore attaches itself to the polar microtubules. The polar microtubules. So if I'm to get one chromosome here and I zoom in, it would actually look like this. This is my centromere, this is my chromatid, this is my chromatid, and this is my chromatid, and this is my kinetochore. Now the polar microtubules shall attach themselves here to the kinetochore protein. Then from metaphase, the cell proceeds to anaphase. We said ana means apart. So these chromosomes shall be pulled apart, but they don't go as chromosomes, they instead go as chromatids. So I'll say here that the chromatids are pulled to opposite ends of the cell. So I'm assuming still this is my cell. These are my microtubules, and these are my chromatids. Now the centromere shall disappear, and this one shall go as individual chromatids. This one shall be pulled this way, and this one shall be pulled that way. Such that at the end of the day, each daughter cell, one daughter cell takes the same number of chromosomes, and the other daughter cell also takes the same number of chromosomes. And this pulling is aided by what we call motor proteins or motor proteins. The motor proteins are the ones that pull the microtubules of this side, and they also pull the microtubules of the other side. Then from anaphase we shall go to telophase. So from anaphase I will go to telophase. Then from telophase my cell shall go back to the G1 phase. So telophase. So in telophase, I'm assuming this is number one, we shall have development of what they call the cleavage furrow. So I'm assuming this was my cell. Now the chromatids here have reached opposite ends of the cell, and the chromatids here have also reached opposite ends of the cell. Yes. Then there will be something in between here that will separate the two, what we call the cleavage furrow, and it usually develops in the place of the metaphase plate. So the cleavage furrow develops in the place where the metaphase plate originally was. That is number one, the cleavage furrow develops. Then number two, now since these chromatids are at opposite ends of the cell, they become less condensed. So they become less condensed to form chromatins. So when they become less condensed, they will go back to the original form of being like thread-like structures. And when they form thread-like structures, they need to be protected from other cellular components. Therefore, my nuclear envelope shall regenerate. The nuclear envelope regenerates. And that is what happens in telophase. And telophase ends with another process called cytokinesis. But before we venture into cytokinesis. I want us to have a brief review of the cell cycle. I'm assuming now this is my cell cycle. We say the cell cycle begins with the gap 1 phase, then the cell shall proceed to the S phase, which is the synthesis phase. Then from there it shall venture into the gap 2 phase. Then from the gap 2 phase the cell shall go into the prophase. Then from there it shall go into metaphase. Then from metaphase it shall go into anaphase. Then from anaphase it shall go to telophase. And we say gap 1 to gap 2 is what we call interphase. And this is the longest period in the cell cycle. Then from prophase to telophase is what we call the M phase, or what we call the mitotic phase. In fact, the mitotic phase usually takes up to 1 hour. We say this one takes up to 8 hours, this one takes up to 6 hours, and this one takes up to approximately 4 hours. So this is generally the cell cycle. And in the cell cycle, we have various checkpoints. So the first checkpoint is from the gap 1 to S phase. From here to here. This is what you call the G1 stroke the S phase checkpoint. Like there are some certain conditions that have to be fulfilled for the cell to cross from the gap 1 to the S phase. If they are not fulfilled, the cell shall enter what you call the G0 phase. The G0 phase. So the G0 phase is when the cell is not preparing to divide. When this signal is not overridden, the cell shall enter a period called the G0 phase, or what they call the quiescent stage. The quiescent stage, when the cell is no longer dividing. However, if the conditions are fulfilled, the cell shall enter the S phase, then the G2 phase. Then from the G2 phase, we have another checkpoint here. The G2 checkpoint. G2 checkpoint. Then the other last checkpoint is at metaphase, what they call the end checkpoint. If not the end checkpoint, other texts call it the spindle checkpoint. The spindle checkpoint. We are going to see what happens at these various checkpoints and what happens if those checkpoints are not fulfilled. So that is it for the cell cycle.